involve evolution. Like, you don't understand like how much we need it as a tool, and it's a functional tool.
It's functional as a model, but if evolution was not true, you
then
could
why
still
does all of our
read
research work?
RNA viruses.
And that's incredibly childish to say, oh, it's a snake, it's always going to be a snake, it's a rat.
I said you could
it's
still read RNA viruses if evolution wasn't true.
But the endogenous retrovirus research would absolutely not work at all, and yet it does.
Well, how many of the endogenous retrovirus sequences are there in a human genome?
There are tens and tens of thousands.
Tens and tens of thousands. Well, how many of them do we share in common with chimpanzees?
It depends on how you count. If you're considering all viruses that are complete, meh. If they're considering all...
What's the range?
As far as we know, complete endogenous retroviruses, I believe there's 12 to 14.
Okay, 12 or 14 of them. And there's 8.5% of our DNA is endogenous retrovirus DNA. Well, what's the chance that out of those 98,000 genes that there'd be 14 orthologs? I mean, I'm not saying that I have proved that that's not evolution, but I am saying it could happen without. Isn't that true?
And we can tell the difference when it happens. That's what we were talking about with the lemurs.
Well, what about the ones that are considered definite common ancestor ones? How many of those are there?
I only know the complete sequences because that's only what...
14 of them?
Yes.
Okay, so there's 14 that line up with the evolution theory out of 98,000.
No, of the complete endogenous retroviruses. These have been in our genome for 30 million years at least.
By the theory,
so
yes.
There's lots of them that are either... They're only the LTRs because LTRs match up and you can pop out that endogenous retrovirus when things don't work perfectly. And then there's ones I talked about that have mutated over time to have various stops and that sort of thing, so they're non-functional anymore. The ones we're really interested in, the ones we've looked at first, are the very young ones that can still code potentially for an endogenous retrovirus particle that can cause a cancer or a disease in people.
Well, I would agree that the 14 that you have go along with the evolution model. But what I'm questioning is, what are the chances that 14 of them would out of 98,000 anyway? Now, like I said, it could be because of evolution. It could, and so there's no other way those 14 could be.
Where a retrovirus inserts into a genome is absolutely random. Some like to insert near maybe more active genes or less active genes, but the actual insertion site...
Gene poor areas, coding poor areas, right?
Not necessarily.
So they are selective to some degree.
Some.
Well, I think we're just leaving dangling this thing that what are the chances that out of 98,000, 14 would line up with evolution? It could mean evolution is true, but it could be a third cause.
The trickster god.
No, not the trickster god. A biological cause. Personally, I don't think that God would, that's just my theology or my philosophy on it, I don't think God would go messing around with those things and trick people any more than you think that he would. I think he wouldn't, you think he couldn't, but, and I'll give you that, that's fine. But the 14 seem to be critical to the argument, are they not?
Yes.
Yes. Well, what I'm saying is they go along with the theory, much like the red blood cells in the T-Rex and the 4 gene pools for all humans go with the creation theory. I think that the red blood cells are good data against an old age for that fossil. I think that the 4 gene pools are good data for it's compatible with the flood model of human population history. It doesn't prove it, but it's compatible. And from what I can see from the 14 out of 98,000, those 14 are compatible with evolutionary theory. Would you say that those 14 are the judge's hammer comes down, it is done, it is finished, evolution is a fact because of those 14?
Personally, like I said, if every fossil remained buried, and if we somehow figured out the technology to sequence genomes without evolution, which I seriously doubt, but if we had, And if we saw all of these sequences and compared them to retroviruses we see now and saw all of these endogenous retroviral fossils, that common descent would have smacked us. It's an obvious conclusion in the year 2000.
So your answer is yes?
Yes.
Okay, so your answer is the 14 are enough.
Yes.
To be a slam dunk for evolution.
The 14
it's the final.
plus all the other leftover garbage that we've subsequently kind of looked at, but not in the...
What's the creationist argument for those 14?
That they were independent.
They got to the same point out of millions of base pairs. How do you explain that they ended up in the same place?
Well, they had 98,000 chances.
No, they didn't. We're talking about millions and millions of base pairs, and they end up, like you said, orthogonal, in the same spots, in different species.
In 4,000 years.
Yes.
They just happen to end up in the exact same spot in chimps and humans?
Yeah, of course the creationist has got to say, well, maybe there's less randomness, even less randomness to the placement of to the insertion of these snippets than is even believed now. It is incomplete knowledge. You must agree that the knowledge is incomplete in Earth research, right?
Yes, and it's only going to be more on the evolution side as we start looking at the things that might not be as important for disease. A lot of research, unfortunately, is not focused so much on just knowing things for knowing things. It's knowing things because it causes a disease. You're more likely to get funding for that.
I want my son, and well, all 4 of my children, to learn the fact. Not the assumption or the theories, what we know for sure.
Well, evolution is a fact, and it is a theory.
But how do we
And
know
we
for a
have
fact
evidence.
it's 30 million? How do we know million?
It's a whole bunch of different lines of evidence that come together. I'm just talking about my specialty, which is endogenous retroviruses. But it's people like me, it's people like the cell biologists, it's people in the geology field. We're all working together and coming to the same conclusions.
Yeah, I'm not talking
so
about
that evolution as a fact.
And evolution, I'm talking about the difference between 1,000,000 and 1,000.
Yep, there's a whole bunch of different lines of evidence, and we're all going together.
If evolution is supposed to be a process, a slow process, one of my questions is, are there species that have irreducible complex systems that have to be in place before they could ever exist? And if there is, how does the evolution explain this?
I'd like to talk about that too, because again, to go back to plants, in undergraduate research, we were working with another lab where they had a gene duplication event, and the second gene, over the course of time, we were watching this gene, which is, this lab went back maybe 25 years. This gene was becoming irreducibly complex before their eyes. Slowly but surely, as they bred these plants, it could do without this gene even less and less. And again, looking at it from a microbiologist perspective, the big thing, of course, is the bacterial flagella. And the scientific consensus is that actually the flagella, and so the reason why bacteria make you sick, not all bacteria make you sick, because like your intestine is full of bacteria, your mouth is full of bacteria. There are certain bacteria that make you sick because they have special genes that are actually given to them most of the time by viruses, which is why I think it's cool. And these genes code for a secretory pathway that allows them to shoot out these toxic proteins that actually really help the bacteria replicate. And so that system is related to the flagella, which is also another pathogenicity marker for bacteria. So the very first thing an E. coli needs to attach to you to make you poop all the time is to have a flagella so it can stick to your intestines, otherwise you just get pushed through. But it also needs these other things that were brought in by viruses. And again, just another pet hypothesis of mine, when you look at these secretory channels, to me they look like a phage tail inverted, so and again, they got it from a virus, they just sort of domesticated it and played with it a bit. I have no proof of that last thing. That's just me being a very virus-centric person. But a lot of things that maybe right now appear to be irreducibly complex weren't so not too long ago. And sometimes, I believe, there was a recent paper where they took out a lot of parts. and they still got a functional machinery. So to say something